Hello everyone, welcome to QXON. In this video, you will learn about pH and buffers. In this topic, I will be covering what is pH, how acids and bases contribute to change in pH, then what are buffers, and with respect to buffers, I will talk about equilibrium constant, Henderson-Hasselbalch equation and physiological buffers. So let's begin. So what is pH? pH is negative logarithm of hydrogen ion concentration, which is expressed as minus log hydrogen ion concentration in big brackets. It is a measure of acidity or alkalinity of a solution. For pure aqueous solution, water molecules dissociate into its components, that is hydrogen ions and hydroxyl ions. And hydrogen ion concentration in pure water is 10 to the power minus 7 moles per liter. With the change in the hydrogen ion concentration, there will be either acidic solution or alkaline solution. So at 10 to the power minus 7 moles per liter of hydrogen ion concentration, pH is 7. If the hydrogen ion concentration increases, which is shown by decreasing negative values, the solution becomes acidic and the pH becomes lesser than 7. On the other hand, when hydrogen ions decrease in concentration, which is shown by increasing numbers, negative values, the solution becomes alkaline and the pH becomes more than 7. So this is the hydrogen ion concentration and this is the pH. Normal plasma pH is 7.35 to 7.45, and with each unit change in pH, that is from 7 to 6, if the pH becomes from 7 to 6, there is a 10-fold change of hydrogen ion concentration. So when hydrogen ion concentration changes from 10 to the power minus 7 to 10 to the power minus 6, that means it has increased 10-fold. pH changes 1 unit, that is it becomes from 7 to 6. Now, any substance which adds hydrogen ions into the aqueous solution is known as an acid, while any substance which removes hydrogen ions from the solution is known as a base. So, here we have shown a water molecule which dissociates into hydrogen ions and hydroxyl ions. So, suppose this is pure water, okay. So, there will be equal concentration of hydrogen ions and hydroxyl ions. Now, suppose there is addition of another substance HA which dissociates into hydrogen ions and its conjugate base. What is happening that this substance is increasing the concentration of hydrogen ions in the solution. So, this will be an acid. On the other hand, if we take another substance which dissociates into hydroxyl ions and its conjugate acid, what is happening that this hydroxyl ions will in turn combine with hydrogen ions of these aqueous solution and will remove that hydrogen ions from the aqueous solution, and the combination of hydroxyl ion and hydrogen ions will produce water. So this is an acid because it is adding hydrogen ions into the solution, and this is a base because it is removing hydrogen ions from the solution. But there is a difference between what strong acids and bases do compared to what weak acids and bases do. These strong acids dissociate completely into respective ions and cause change in pH. Let's see an example. So, HCl is an example of a strong acid which dissociates completely into hydrogen ions and chloride ions and adds hydrogen ions to the solution. On the other hand, NaOH, sodium hydroxide, is a strong base which again dissociates completely into sodium ions and hydroxyl ions, which in turn combines with the hydrogen ions as already told and forms water, creating a deficit of hydrogen ions. So, strong acids and bases basically dissociate completely into respective ions and cause change in pH. Now, in our body, the cells are continuously adding some acids and bases into the body fluids, and our food also, some acids and bases are being added due to that. So, these acids and bases will tend to change the pH of our body fluids. But if that happens, that can be disastrous if it goes beyond a certain limit. Why? because the enzyme activity and the protein structure depend on pH. So, the change in pH is prevented by something known as buffers. So, let's take an example. Here is the acid which is dissociating into its components. Now, when we talk about buffers, they also dissociate into its components. So, here BH is dissociating into B minus, the anion and hydrogen ions. And let us see one base also, say suppose sodium hydroxide, which is dissociating into sodium ions and hydroxyl ions. So, what will happen that this base, this buffer which we are talking about, it will prevent the change in pH that is being caused by the addition of either a strong acid or the strong base. So, how that will happen? See, this buffer which we have shown, though it is dissociating into components, but it is not dissociating completely, which is different from what we spoke about strong acids and bases. So, this buffer is actually either a weak acid or a weak base. So, the one shown here is a weak acid. You see the arrows, they are pointing in both the directions. That means for a buffer, the reaction can go either in the forward direction or in the backward direction. So, let us see that how this buffer actually acts. So, you see that this anion of the buffer can combine with the hydrogen ions which are being added due to the addition of an acid, and then the reaction will proceed in this direction. So, this BH will form, the compound will form if the acid is added into the solution. On the other hand, the BH can combine with the hydroxyl ions which are being added due to the addition of the base, and the reaction will proceed in the forward direction, forming its components, that is the anion and the hydrogen ions. So, fundamentally the action of the buffers depends on this bidirectional dissociation. So, either the reaction can proceed from BH to formation of its ions, or the ions can combine and form back BH.